Today we are going to start the revision series on our special senses, physiology. So first we are going to start with hearing, olfaction and taste. So mainly now let us start with ear first. So ear has mainly two functions, that is the main thing for ear is of course hearing, and that is done by our cochlea. Cochlea is present especially in our inner ear. So ear is divided into outer ear, middle ear and inner ear. So inner ear mainly have cochlea, which is useful for hearing. And next, balance. Balance is done by our vestibular apparatus, and this vestibular apparatus is also in our inner ear. And remember that this both together by mainly cranial nerve 8, that is our vestibulocochlear nerve 8. This you can say means this both are like together, vestibular apparatus, cochlea, from the vestibulocochlear nerve. Vestibulocochlear nerve. So useful for both balance and hearing. So ear's main two functions, balance and hearing. Let us see the main function of hearing of our ear. So mainly two membranes divide the cochlea into three parts. So here you can see this is our cochlea. So two membranes are there which are dividing the cochlea into three parts. The two membranes are, you can see the lower membrane, the lowermost membrane is a basilar membrane, the upper membrane is a resinous membrane. So basilar membrane, resinous membrane, dividing the cochlea into scala vestibuli, scala media, scala tympani. So there are three parts in a cochlea. So next next stria vascularis. So you can see here this is stria vascularis, which is a extra lining for our scala media. So this is mainly releasing the endolymph. So remember, endolymph is present in the scala media and perilymph is present in scala vestibuli tympani. So what is this endolymph? Perilymph is the fluid which is present in it, and the endolymph leads to formation of endolymphatic potential, that is of plus 80 millivolts. So why here the positive charges? Because of more potassium. The more potassium is released by means of our stria vascularis layer. This layer releases more potassium. So because of higher secretion of potassium, this is causing a potential of plus 80 millivolts, and that is called as endolymphatic potential, because here endolymph is present, so forming an endolymphatic potential of plus 80 millivolts. And it's like just a cell membrane. See, inside the cell we have most potassium, outside the cell we have more sodium, right? So outside the cell, if it's outside, it's perilymph, outside more sodium, like in scala vestibuli, scala tympani. Inside the cell we have more potassium, that is scala media. So like that you can live with endolymph. So next, you can see here the scala media contains receptors for hearing. So this scala media, if we zoom inside, you have some receptors for hearing. Those are called as hair cells. So you can see these are the hair cells. So mainly we have two types of hair cells. One is called as outer hair cells, another is called inner hair cells. And these hair cells are nothing but called as organ of Corti, which is a receptor for hearing. Mainly the receptor for hearing is present in the scala media of our cochlea, where there are outer hair cells and inner hair cells. And these two cells should be connected each other by means of dater cells, and these dater cells are the supporting cells which are connecting both the outer hair cells and inner hair cells. So you can see this, the outer hair cells are like having more number, means they are more in number, and but they are innervated less. But inner hair cells are having less in number. They are less in number but highly innervated. So by which nerve they are innervated? They are innervated by the cochlear division of cranial nerve 8. So cochlear division of cranial nerve 8 innervates both outer hair cells and inner hair cells, but inner hair cells is highly innervated and outer hair cells is less innervated. And next here you can see outer hair cells are more prone for damage by ototoxic drugs such as aminoglycosides. So example, you can take streptomycin. So by usage of this kinds of drug, there is some damage, more damage for outer hair cells, because they are numerous and also they are present outside mostly. So these drugs can act upon it. Example, streptomycin. And the receptors which are innervated by cochlear division of cranial nerve 8 are both outer hair cells and inner hair cells. You can see inner hair cells are less numerous and highly innervated. Outer hair cells are more numerous, less innervated. Already discussed. Now let us see about hair cells in somewhat detail. The tallest cilia on the hair cell is called gynoecilia, and the hair cells adjacent to it are progressively decreasing in height and called stereocilia. So for example, I will draw it. So these are like hair cells. So here the tallest one is called gynoecilia and the smaller one is called stereocilia. So hair cells bend towards gynocilium. So if the hair cells are moving like this, these are the small hair cells called stereocilia. So if the stereocilia are moving towards the gynocilia, then the process, you can see here, causes depolarization. So depolarization is input of positive ions like especially sodium, or you can take here potassium influx from the endolymph. So this leads to signal transmission. So this is all taking place especially in our scala media. In the scala media only we have hair cells, right? In that hair cells only again we are having some hair cells which are small called as stereocilia, and some hair cells are long called as kinocilia. And if this stereocilia is moving towards the kinocilia, it will lead to depolarization mainly because of potassium influx. Why potassium influx? Because you can see here endolymph is present where potassium is secreted more, so potassium influx. And if hair cells bend away from kinocilium, so if they are bending away from the kinocilium, then it will lead to hyperpolarization. So it's a signal inhibition. So if hyperpolarization, signal inhibition. If depolarization, signal transmission. Very important point. And the potassium current measured from outer hair cells is called as autoacoustic emission, OA. What they're saying, potassium current measured from outer hair cells. So this outer hair cells, from this we will try to measure the potassium current, and that is called as autoacoustic emission. And this is also called as Kemp waves. Why? Because the Kemp is a scientist who have found them. And the sensitive test for hearing, this is actually a method of sensitive test for hearing, because the potassium is the one which is leading to endolymphatic potential, and also potassium is the one which is leading to depolarization and signal transmission. So here potassium is playing a very important role. So we need to measure the potassium current especially from the outer hair cells. This process is autoacoustic emission, OAA, and this is also called as Kemp waves. So this is a main sensitive test for hearing. And next we have a traveling wave theory of Becquerel, because Becquerel is the one who found this traveling wave theory. So the sound waves travel in basilar membrane. Actually this is the basilar membrane. As the sound waves travel this on this, they vibrate. So because of this vibration, here the cochlear nerve joins, and to the central nervous system the information goes. So this is the main mechanism of hearing. So you can see here the traveling wave theory of Becquerel. The sound waves travel in basilar membrane. The sound wave causes vibration in basilar membrane depending on the frequency. So according to the frequency of the sound which is coming, that basilar membrane vibrates according to it. So if there is higher frequency sound are perceived better at the base of cochlea, and the low frequency sounds are perceived better at the apex of cochlea. Very important point. So when a high frequency like loud noises, high frequency sound, they're better base of cochlea, they're better heard at base of cochlea, and low frequency sounders are better heard at apex of cochlea. Very important. Now let us see the stapedial reflex. What is it? Stapedial is actually muscle in our ear. What it is doing, let us see. So whenever there is loud noise like about 160 decibels, then it is perceived as just only 120. Means you can see our ears have a capacity to decrease the sound. Even though outside the sound is very high, very loud, our ear only controls them by decreasing it decibels to 120. So sound is nothing but attenuated to prevent the damage to tympanic membrane. It's a protective reflex. So it also a type of attenuation reflex, that is it's decreasing the decibels. So the contraction of stapedius muscles, retraction of tympanic membrane. So actually what happens here is whenever there is lot of sound, then immediately the stapedius muscle will undergo the contraction. So because of this contraction there is retraction of tympanic membrane. And the efferents like cranial nerve 8 especially goes to superior olivary nucleus, which is the center. From the center the efferent comes again cranial nerve 7. This time cranial nerve 7 comes, that is our facial nerve. So this is the main cause for the contraction of stapedius. So actually this is the process of a stapedial reflex. What happens is whenever high sound is coming, then immediately this efferent cranial nerve 8 goes to the superior olivary nucleus saying that high sound is coming, and this will listen to this cranial nerve 8 and it will send a cranial nerve 7 to contract the stapedius. So as a stapedius contraction takes place, there is retraction of tympanic membrane and attenuation takes place, like 160 decibel is converted into 120 decibel. So the clinical significance is lesion in cranial nerve 7. So for example, if there is lesion, of course there will be loud noise is not attenuated, so the person will read the loud noise, so his tympanic membrane is at danger now. So this process is called, this clinical significance, this clinical case is called hyperacusis. This is C, hyperacusis. Let us see the auditory pathway. So in auditory pathway mainly we have a mnemonic, you can remember like ecolima. So ecolima, what is it? E for 8 cranial nerve, C for cochlear division of cranial nerve 8, and O for especially superior olivary nucleus, the process which processes the direction of sound, and L for the lateral lemniscus, and and I for the inferior colliculi, which is useful for process of direction of sound. So you can see the superior olivary nucleus also is useful to process the direction of sound, like from which direction the sound is coming, you are able to listen like from which direction. So that is mainly the function of superior olivary nucleus. Along with that, even the inferior colliculi works with it to process the direction of sound, but it's not later. But the important one is the superior olivary nucleus. And next, M for medial geniculate body, which is a thalamic nuclei. Actually we have this medial geniculate body, and also we have the lateral geniculate body. The lateral geniculate body is mainly useful for vision, whereas medial geniculate body is mainly useful for the auditory, that is for hearing. And the auditory cortex, which is located in the superior temporal gyril, that is the temporal cortex. So these are all the things which are required for hearing of auditory pathway. And you can see the superior temporal gyrus, which is corresponding to Brodmann areas. Already Brodmann areas, right, in somatosensory cortex we discussed. So Brodmann areas 41 is mainly belonging to the superior temporal gyrus. And next, this superior temporal gyrus is also known as Hertzler's gyrus. So now let us see olfaction. So the receptor, now we have completed about our hearing process. Now we'll go to smell process, olfaction. So olfaction, the receptor for smell we have in the roof of nasal cavity, especially on the olfactory epithelium. So the receptor is a type of mainly bipolar neuron. So this is a kind of bipolar neuron, and one end is in the nose region. So you can see one end, the roof of nasal cavity, and another end is reaching the brain, which pierces the cribriform plate. So you can see this is the other end which is reaching towards the brain by piercing the cribriform plate. And this is an olfactory nerve. So olfactory bulb. Olfactory bulb has mainly 4 cells, that is mitral cells, tufted cells. These both cells are mainly excitatory. So as it's excitatory, its neurotransmitter is glutamate, whereas the periglomerular cells and granule cells are inhibitory, and its neurotransmitter is GABA. So you can see we have mainly 2 excitatory cells and 2 inhibitory cells in olfactory bulb. And olfactory bulb mainly lead to in the thalamus, it will reach the dorsomedian nucleus. From there to olfactory cortex, that is orbitofrontal cortex, OFC in short form. From there to discrimination of smell. So how we can discriminate the smell, that this is this kind of smell, mainly by this process. The olfactory bulb is actually in the CNS. From there it will go to thalamus, especially in the dorsal median nucleus of thalamus. From there to olfactory cortex, that is orbitofrontal cortex. From there to, of course, we can discriminate the smell. And next we have the limbic cortex, which is also called as smell brain. So it is divided again into 2 parts, like endotorcinal cortex and amygdala. So endotorcinal cortex is where the smell and memories are processed together. So whenever you see the smell, you will remember that this kind of person have this kind of smell or perfume, like that. And the degeneration causes Alzheimer's. So actually in Alzheimer's, the person will have the degeneration of the smell and memories of especially this entorcinal cortex. So the person can't see the perfect smell and think of the memories associated with it. And amygdala is also smell and emotional process. So this is about this olfactory bulb. And next let us see, olfaction has the smallest range of intensity. So what they mean to say is intensity discrimination. So actually olfaction is seeing smell. If you are seeing smells of different things, you will see that you can see, means you can only smell that, but you can't see that how much intensity the smell is coming, whether it's higher or lower. Or humans won't usually keep that much focus on that. They see the smell and they'll not think how much smell is coming. So that's why olfaction has smallest range of intensity discrimination. And next let us see taste gustation. So the receptors for gustation taste is mainly are, of course, taste buds on our tongue. So the taste sensations, mainly we have this 5 basic tastes: sweet, salt, sour, bitter, and umami. So what it is umami? Umami is a most addictive taste. It's mainly due to a chemical called monosodium glutamate, which is in a short form called MSG, which is called as also Ajinomoto. So because of this, there is addiction of food. So these are all the 5 tastes which a human can taste. And next we have the taste buds innervation. So taste buds innervation is mainly done by cranial nerve 7 branch chorda tympani. especially in anterior two-third of tongue, and from the posterior one-third of tongue you have the cranial nerve 9, that is glossopharyngeal, and especially from posterior most and palatal region of tongue you have cranial nerve 10. So cranial nerve 7, 9 and 10, 3 together relay in the medulla. In the medulla mainly they reach the nucleus structus solitarius, which is a taste integration center. So you can see we have wrote it in short form. Why? Because N for 9th cranial nerve, T for 10th cranial nerve, and S for 7th cranial nerve. So you can see all 3 cranial nerves relay in this structus solitarius for taste integration, and later they will go to the ventropostromedial nucleus, that is VPN nucleus, which is present in thalamus, and from there from the thalamus they go to the taste cortex, especially which is located in the insula part. So this is all about it. You can see like even for the taste, even for the smell, even for the vision, everywhere the relay center thalamus is present. So that is a common thing you need to observe. See, for the taste pathway you have thalamus, and in your auditory pathway also you will have your thalamus, like medial geniculate body, which is a thalamic nuclei, and even in your vision you will have your thalamus, and even your smell olfactory bulb, you can see you can have your thalamus, especially dorsomedian nucleus. So these are all you need to remember about the special senses. And next we'll move on to vision, which is the last part of our special senses revision. Revision series on vision. So mainly we have, first we need to understand the visual neurophysiology. So vision is a special sense, and it has main 3 control points, that is a receptor. The receptor is a photoreceptors, of course, which are present in our retina, and the second one is a thalamus, which has especially a nucleus called lateral geniculate body, and the third one is a visual cortex. Of course it's in our cortex. So these are the 3 main cortical control points. First we need a receptor, that is a photoreceptor in the retina. So as the light passes first to the receptor in the retina, next it goes to the thalamus nucleus, especially lateral geniculate body nucleus, and from the nucleus finally it will reach the cortex. So this is how the light entering will reach finally to the brain. So the role of retina in vision is, retina mainly functions the phototransduction. So what is this phototransduction? Let us understand. So retina has mainly 7 cells, and among that the 6 cells only have the role in vision, and the 1 cell is exception that it doesn't have any role in vision. So let us see what are those 6 cells which have role in vision and 1 cell which doesn't have any role in vision. So mainly the cells in retina, especially the first cell is rod and the second cell is cone. Among the 6 cells, the first one is rods and second one is cones. So rods are more in number, like about 100 millions, whereas cone are just in 5 million. And according to the vision, the photoreceptors of rods are mainly present during dim light, that is they are useful especially in night vision, whereas cones, photoreceptors for daylight and color vision. And the rods are evident in peripheral retina, whereas the cones are mainly present in central retina, that is foveola, which is a rod free region. And next, the rods, they are mainly neurons, and even the cones are neurons. And the rods neurotransmitters are mainly glutamate, and even the cones, the neurotransmitter is glutamate. So now let us see bipolar cells. If you remember, even in our olfaction we have our bipolar cells. So these bipolar cells are the one which receive the information both from the rods and cones and transmit it to the ganglion cell. So the next cell is ganglion cell. So these are all the cells which are in a flow of 6 cells which have role in vision. So first one is rod, second one is cone, and the third one is bipolar cells, and the fourth one is the ganglion cells. So ganglion cells are the only output cells of retina, and the axons of ganglion cells from the optic nerve only cell to produce action potential. So you can see it's the only output cell of retina which is coming out from the retina, and the axons of these ganglion cells from the optic nerve is the only cell to produce the action potential. So from the optic nerve, these ganglion cells take the action potential and travel it towards the brain. And next, the fifth cells we have in our retina cells which are useful for vision are horizontal cells. So mainly these are the adaptation cells. They adapt to brightness of light and connects the rods and cone. So remember, horizontal cells, so as they're horizontal just like a line, they connect the rods and cones together, and they adapt even to the brightness of light. Like for example, if you're coming suddenly from dark light to bright light, so that adaptation is also mainly due to horizontal cells. And next comes a sixth cell, that is our amacrine cell. So amacrine cells are also adaptation cells which connect the bipolar cells and ganglion cells. So the bipolar cells From there only it is transmitting to the ganglion cell of information of rods and cones, right? So the connection between both is mainly due to hemicrine cells. So horizontal cell connects the rods and cones, hemicrine cells connect the bipolar cells with the ganglionic cell. So mainly the neurotransmitter of hemicrine cell is acetylcholine. Next we'll see the retinal glial cell. So this is a 7th cell, as we said, total 7 cells in retina, and this is the 7th cell which is actually not having any role in the vision, so no role in vision. It's only a supporting cell, that is a retinal glial cell, and this is also known as Muller's cell. So now let us see the process of phototransduction. So mainly the anterior layer of retina ganglionic cell layer, in the presence of light stimuli, it converts into posterior layer photoreceptors. So you can see the phototransduction process, the anterior layer of retina ganglion cell layer in the presence of light stimuli to the posterior layer which is photoreceptors, and 2 states of retina, dark and light. So rhodopsin is also known as the visual purple pigment, and the rods mainly contains rhodopsin, which is a combination of both opsin and vitamin A, and vitamin A is especially in the retinal form, which is an aldehyde form, and in dark state the 11-cis retinal is converted to all trans retinal by light. So whenever in dark, the 11-cis retinal is converted into trans retinal in light. So in light it is in the form of all trans retinol, whenever it's in dark, 11-cis retinal. So now let us see dark state rods. So as you have seen, 2 states of retina, dark and light. Now we will see the dark state rods. Rods have resting membrane potential of -60, mainly due to potassium efflux, and rods in dark state have higher amount of cyclic GMP. So because of this they open the sodium channel and sodium influx takes place, and this is because of this causes depolarization. So this sodium influx is called as dark current because it's in a dark state. So what happens in dark state? The resting membrane potential first is -60, later the because of increase in cyclic GMP, sodium channels open, sodium influx, so there is depolarization. So in dark state the 11-cis retinal is present. Very important, as we discussed before, dark state 11-cis retinal is present. Now let us see light state rods. So in rods we are seeing 2 cases. When the rods are in dark state there is depolarization. Now let us see light state rod, what happens. So 11-cis retinal, of course in the presence of light, is converted into all trans retinol, and it's activating the GPCR protein, that is G protein coupled receptor, and this activates the transducin. So because of this activation of transducin, it activates the phosphodiesterase, and this phosphodiesterase does the hydrolysis of cyclic GMP. So because of this the sodium channel closes here, so it's opposite. So in a light stage the sodium channel is closing and leading to hyperpolarization. So remember, in light stage of rods there is hyperpolarization and there is all trans retinol. In dark state there is 11-cis retinal and also there is depolarization. And dark stage depolarization and light stage hyperpolarization, that's what we are discussing about the rods especially. And this is true for all cells except hemicrine cells. Hemicrine cells are purely depolarized both in dark light. Very important point. So hemicrine cells are the one, if you remember, they connect the bipolar cells to the ganglionic cells, and only in these cells, either in dark stage or light stage, there is always depolarization. But in our rods, during dark stage there is depolarization, D for D, and light stage there is hyperpolarization. Now let us check out the Wall's visual cycle. So you can see here the dark stage in the night and the light stage in the day. Of course, this conversion of 11-cis retinol into all trans retinol is a Wall cycle whenever in light stage, or conversion of this all trans retinol into 11-cis retinol in the night stage or dark stage. So this cycle is Wall's visual cycle. Now let us see the funny currents. So they are the sodium channels which open in the dark stage and close in the light stage. Yeah, of course, if they are closed in light stage there is hyperpolarization. If they are open in dark state, that it will lead to depolarization. So they are seen in eyes and also in a SA node. If you remember, in a heart physiology also we discussed about this funny currents for heart rate generation. So mainly for heart rate generation also they are useful. And these are also known as HCN channels. There are hyperpolarization cyclic nucleotide channels. So funny currents are also called as hyperpolarization cyclic nucleotide channels, that is in short form HCN channels. And let us see about ivabradine. Ivabradine, funny current blocker. So ivabradine is a funny current blocker. It mainly decreases the heart rate and also it is used in arrhythmia. So in the name itself you can see ivabradine, brady like bradycardia. So as here there is higher heart rate, to decrease it of course we need to break into bradycardia. So you can remember like that, ivabradine. And now let us see the lateral geniculate bodies. So lateral geniculate bodies mainly the light which is coming to the retina, from the retina it goes into the thalamus, and the message which the light when it hit the photoreceptors, it will send the message first to the retina. From there it will reach the thalamus. Especially in the thalamus it will reach the lateral geniculate body, and the lateral geniculate body it mainly has the 6 layers, and the 1, 2 and 3, 4, 5, 6. 1, 2 layers are mainly magnocellular layers, and it leads to magnocellular pathway. And because of this magnocellular pathway, there is movement of eyeballs, that is especially extraocular movements. And 3, 4, 5, 6, there is parocellular layer, that is leading to parocellular pathway and mainly useful for the color vision. So you can see here the distribution of fibers among the lateral geniculate body. So remember, lateral geniculate body have 6 layers, right? So 1, 2, 3, 4, 5, 6, and the layers 2, 3 and 5 are ipsilateral. Ipsilateral means they are coming from the same side, but the layers 1, 4, 6 are contralateral. Contralateral means they are coming from the opposite side. So let us understand what is this opposite and ipsilateral and contralateral. So first we'll see the contralateral. So for example, the left eye and the right eye. From the right eye you can see the 1, 4 and 6 which are dark in color. These are mainly contralateral because these are all on the left side, but from the right eye it is crossing towards the left side and coming, so hence they are contralateral. Whereas 2, 3 and 5 they are coming on the same side. This is light one, they are coming on the same side from the left eye, so they are ipsilateral. So these are the 6 layers of lateral geniculate body which is present in our thalamus. Now let us see the visual cortex. So visual cortex is from the lateral geniculate body to the visual cortex. The location is mainly occipital lobe. So this visual cortex is present especially in our brain in the occipital lobe, and the Broadband area is according to 17, 18, 19, that is calcarine sulcus. And the calcarine sulcus is anatomically mainly contains the myelinated fibers, and these are arranged like strips or striations in the visual cortex. So these are also called as striate cortex, or also known as striae of Jennari. And it contains the simple cell and complex cell, and identify the individual features called feature detectors, blobs, and useful for mainly color vision, because we can identify different features of course in the presence of color only, so for useful for color vision. Now let us see the color visual pathway. So mainly whenever the light hits the cones in the retina, so it sends the information to the thalamus, especially to the parocellular pathway, and from there it will reach the visual cortex, especially the blobs. And the cones are having 3 types of primary colors it can find out, that is red color, blue color and green color. For the red color it is called L cone. If absent it will lead to protanopia. And remember, L means here long wavelength, and S means here short wavelength for blue color, and M means here medium type of wavelength for green color. So if blue color is absent, then it is called trinopia, means if a person is not able to see blue color, trinopia, and if a person is not able to see green color, then it is called deuteranopia. So these are the things we need to remember. So these are the 3 basic colors by which we can by cones we can see. And color blindness. Color blindness is the most common defect, where there can be red-green defect, and it is mainly X-linked recessive disorder, and females are the carriers here and the males are the affected person. So next, striate cortex in the occipital lobe, extra striate cortex, parietal, dorsal pathway, extraocular movements, temporal ventral pathway, color vision. So what they are saying, striate cortex in the occipital lobe. Actually the striate cortex is present in the occipital lobe. Other than the striate cortex, we have some extra striate cortex, that is parietal, dorsal pathway, extraocular movements, and in temporal we have the ventral pathway which is useful for the color vision. And Meyer's loop, temporal cortex visual fibers, Baum's loop, parietal cortex visual fibers, and the visual information from the face region is going to the temporal cortex, and it will lead to face identification or recognition, and especially in the inferior temporal cortex. So especially in the inferior temporal cortex, by help of this we can recognize the face of an individual. Clinical correlation is prosopagnosia, that is due to lesion of inferior temporal cortex. So whenever there is any lesion in the inferior temporal cortex, a person cannot identify the face. It's a difficulty in face identification. Next, let us see the visual pathway, the order of neurons. The first order neuron in the visual pathway is photoreceptors, that is the rods and groans, and here the neurotransmitter is glutamate. And the second order neurons is bipolar cells, means from the photoreceptors it will go to bipolar cells, from there it will go into ganglion cells. Of course, from the ganglion cell the optic nerve is connected. And the fourth order neuron is lateral geniculate body in the thalamus. From the lateral geniculate body finally it will reach the visual cortex, which is the fifth order neurons. So these are the different orders of neurons which are seen in a visual pathway. So we need to remember all this. So by this we completed our special senses complete revision.